Welcome to this course on organic chemistry and biology and drug development. In the last session or the last few sessions, we were covering or we are studying the antimicrobial agents, and we have seen the chemistry of penicillins, how do they work, and then alongside other various kind of bacteriostatic as well as bactericidal antimicrobial compounds, their chemistry has also been told. Now, I will start with this slide which basically represents the all spectrum of antibacterial agents that we have and their target of action. Like here this is the bacterial cell, and the bacterial cell wall is so important that we know now. So, what works against the cell wall? All the beta lactam antibiotics, then the glycopeptides which include vancomycin and bacitracin. Okay. Then you have some antibacterial agents which work against the DNA forming DNA replicating enzymes or transcription process which hampers the transcription process. So, we say that the target is DNA synthesis or RNA synthesis, like the fluoroquinolones, the DNA gyrase inhibitors. DNA gyrase is basically a kind of topoisomerase which takes care of the supercoiling problem when the DNA strand unfolds, like ciprofloxacin, levofloxacin, moxifloxacin. RNA synthesis is stopped by rifamycin or rifampicin, anti-tuberculosis drug. Then plasma membrane, there is a class of compounds polymyxin and lipopeptide, and then there is this ribosome. This is a big class of compounds which targets the ribosome. It could be 30S subunit, could be 50S subunit, aminoglycosides. Then you have like amikacin, tetracycline, macrolides like erythromycin, okay, chloramphenicol, etc. And then you have metabolic pathways like which hampers the metabolism, like folic acid metabolism, and you have the sulfonamides, sulfones, trimethoprim. And then in tuberculosis you have mycolic acid biosynthesis. We have not touched that, we have not talked about this mycolic acid, that is a special that itself is a special topic, that how to stop the growth of the tuberculosis microorganism. But what they have is basically in tuberculosis the lipid layer is made up of not C20 or C16, C18 amino acids, fatty acids like the stearic acid, palmitic acid. It is not like that, which is present in all other bacteria, but in tuberculosis the lipid bilayer is made up of extremely long 60 to 70 carbon atom containing fatty acids, and they are extremely difficult to penetrate. Penetration will be very difficult because it is a thick layer of hydrophobic membrane containing, it is a bilayer containing about 60 to 70 carbon containing fatty acids. Okay. These are called mycolic acid. So, if you can stop the mycolic acid biosynthesis, which many of the tuberculosis drugs do, so that is like isoniazid. It is a very simple compound, but it is a pyridine based compound. We have not talked about that, just to let you know that mycolic acid is a special class of lipid molecules, and they are very difficult to penetrate. So, all these other antibiotics do not work against the TB organism. So, you have to stop the mycolic acid biosynthesis, so that it is not formed, and isoniazid is one of the very well known anti-tuberculosis drug. Okay. Now, last time we ended, we took up a topic which is called the engineered antibiotics or engineered antibacterial agents. That means, where you manipulate the protein that makes the antibacterial compounds. specifically the antibiotics, because now we are talking about proteins, that means they are made by the living organism. Okay. So, now we started with this engineered antibiotics, okay. That means change the structure of the antibiotic. We started with penicillin, and to know that, to do that engineered antibiotics, what we need to do is to know how the bacteria first makes that antibiotic, and then you then modulate the different enzyme systems that are involved in the biosynthesis of that antibiotic. Okay. Now, remember, there is enzymes which ultimately makes the antibiotic. Okay. So, you can target new antibiotics by changing or by forcing the enzyme to accept modified substrates. Okay. That is one strategy that you take at the protein level. You can dictate the protein to accept modified substrates, because we know in penicillin there are 3 amino acids which combine to make a tripeptide, and that tripeptide undergoes a cyclization by a single enzyme, double cyclization by a single enzyme. Okay. And so, if you can change the tripeptide, say the tripeptide is actually LLDACV. LLDACV, A stands for alpha amino adipic acid, then C for cysteine, and your ACV, V for valine. Okay. Now, if you change valine, put some alanine there. So, ACE, the question is whether that will be accepted as a substrate by the enzyme. If it is accepted, then you will get a penicillin where 2 methyls are not present, only 1 methyl. Okay. So, that is one way, that giving modified substrates to the proteins that are involved in the biosynthesis. That was the old strategy, that is not the new strategy. The new strategy is that rather than going for the protein, because the protein at some point of time, especially in the 80s or 70s, it was a very precious item, because ultimately if you try to isolate the protein, it will be very few amount, means it is a very few quantity, micrograms. To get milligrams of protein is a challenge at that time. Now, what people do, it is because see this approach is restricted by the ability of the enzyme to accept modified substrate, because if alanine is not accepted as a substrate, then you are not getting the penicillin which you wanted to get. Okay. So, it all depends how specific is the protein. If it is too specific, very narrow substrate dependence, then you will not be able to get newer modified penicillins. On the other hand, if it is really have a broad specificity, then you can change the tripeptide, put different amino acids, and then you can get new molecules. Okay. But again I repeat, this was the earlier strategy. Today what people do is they actually do the reverse engineering. So, they see this protein is involved in biosynthesis of antibiotics, they go for the gene, that what are the genes that are involved. What are genes? Genes are the functional part of the genome. Okay. So, there may be several genes which may be, see this gene expresses one protein, say A, this gene another protein like B, and this third one again produces C. Okay. Now, what you do, if you can change at the genetic level, which is these days is quite easy after the advent of polymerase chain reaction, and then now the latest technology of gene editing, CRISPR-Cas9 is a system which is where you can edit the gene. So, if you edit the gene at the genetic level, then what happens? If you put the gene in a live microorganism, then you always get the modified, whatever modified systems it will produce, you are going to get. Okay. So, there are two approaches, you can approach from the protein side, or you can approach from the DNA side, the gene. Okay. I will show you both the approaches. First, it is the proteins that are, we are just discussing about the proteins, no DNA we are discussing here. In case of penicillin, what happens? Last time I said that penicillin requires basically this assembly of the 3 amino acids LLD ACV, and that LLD ACV then cyclizes in a single step. Okay. Single step, and you also we told you have been told that what is the rate determining step. See, this is your tripeptide, that is the ACV, SH, and we are not writing the, here those are the alpha hydrogens and then CO and then R. Okay. Now, you know the enzyme that cyclizes this is called IPNS, isopenicillin N synthase, and that synthase, okay, it does not require any ATP. There is a co-substrate requirement, oxygen, and there is a cofactor requirement, that is ferrous, and to keep the iron in the ferrous state you need ascorbate. However, another enzyme which also gives better yield of isopenicillin N by this enzyme is what is called catalase. I will tell you why catalase is added, why does it improve the yield. First of all, let us look at the reaction. The reaction gives you straight away this, the penicillin. Okay. But this penicillin is what is called the isopenicillin N, because it has a L alpha amino adipoyl side chain. So, this is called isopenicillin N. Okay. These are alpha, okay, hydrogen, hydrogen. What is the other product? What is the status of oxygen? This comes as 2 molecules of water. You are removing 4 hydrogens, one is here, one is here, another is this, and another is the sulfur hydrogen. Okay. So, the question, first question was which bonds. I told you about the kinetic isotope effect experiment that demonstrated that it is the carbon nitrogen bond that is formed first, followed by the carbon sulfur bond. Okay. Out of these 2 bonds, it is more difficult to form the carbon sulfur bond, because this carbon is not functionalized. This is a hydrocarbon side chain. So, the reaction chemically, to do a reaction is very difficult, but enzyme does it, okay, because enzyme has a different approach to do reactions which are chemically very difficult to do. Okay. Now, it has been shown that this water oxygen comes from, the oxygen of water comes from the molecular oxygen that is used in the reaction. It does not come from any water. The source of oxygen is molecular oxygen. Now, this type of enzyme is called oxidase, means where the oxygen is not incorporated in the substrate. There is no incorporation of oxygen in the substrate. If you see the incorporation of one oxygen in the substrate, like suppose if you have RH and you have an enzyme which utilizes oxygen and you get ROH, then that will be called that enzyme will be called monooxygenase. Monooxygenase, because out of the 2 oxygen atoms here, one ends up here and the other ends up as water. And if it happens that see 2 hydrogens are there, if both the oxygens are incorporated in the substrate OH, suppose OH, R is just a general group. So, if both the oxygens are incorporated in the substrate, that is called dioxygenase. So, penicillin is not a monooxygenase, it is not a dioxygenase, it is just, if nothing happens, then it is called oxidase. It can be called desaturase also, desaturase. Desaturase means you are actually desaturating it. Saturation means addition of hydrogen and desaturation means taking out the hydrogen. So, you are taking 4 hydrogens. So, that is desaturation. Okay. Now, what is the mechanism of this reaction now? Now, we know that which is the first bond that is formed, maybe I can write here. Now, the mechanism that has been now quite well established is that you have this side chain R and this is NH, sorry, this is not S, this is S, and then you have this CO2H and you have methyl. Okay. So, the first step that happens is the iron which is bound to the sulfur, okay, Fe, and then iron is having other coordinate because it has got 6 coordinations. So, there may be some water, some may be histidine, okay, but we are not writing all those, okay, histidine, some may be water, but it is in the +2 oxidation state. Okay. Now, what happens? You have the oxygen. You know, oxygen is a triplet, is in the triplet stage. So, 2 electrons, 2 separate electrons, it is a triplet oxygen. So, now what will happen that this is Fe, we are not writing the other arms, this reacts with the oxygen. Okay. This reacts with the oxygen and forming this cyclic diocetane system. Okay. That means, iron is giving 1 electron to this oxygen and oxygen is giving 1 electron. That means, this bond, one of the electron has come from iron, one from the oxygen, and this bond also one of the electron has come from oxygen and one of the electron from the iron. So, iron has now donated 2 electrons. So, now this will be in the +4 oxidation state, okay, because the iron has already involved its electrons to form these bonds. Okay. So, NH and then CO2H this, okay, NH and that. Now, what will happen? The sulfur lone pair comes here, because this is a strained system. So, that now breaks. Okay. So, so you have to form the beta lactam, and what was people thought that it must be a kind of double bond here. So, that because if you can generate this type of system, then you can get a cyclization like this 4 exotrig. So, that you have to generate. Okay. So, this is now S double bond Fe and then OOH, this will take a proton. Now, this you have to also think of the what is the oxidation state of the iron. See, iron is not gaining any electron nor losing any electron. So, iron is still in the +4 oxidation state, and this is NH and that is CO2H, then methyl and that part. Okay. So, night sulfur is plus now. Now, what will happen? One of the hydrogen will come here, that will go there, this goes out as water. Okay. And that forms double bond S. See, that is how the thioaldehyde, see C, there is a hydrogen here, C double bond S H. There are so many aldehydes you can see, but it is very difficult to isolate a thioaldehyde. Thioketones you can isolate, but thioaldehydes are extremely unstable. So, what nature has done? It has not generated any free thioaldehyde. Remember, I told you at that time that when there is a debate which bond is formed first, they tried to prepare an intermediate containing an SH in a 4-membered ring, but that was not stable. Okay. So, that was not stable. So, nature bypassed that by putting the sulfur attached to the iron, okay, so that it stabilizes. Now, the sulfur has a positive charge, and iron, what is the status of this? Iron Fe double bond O, and iron is still in the +4 oxidation state. This is called a superoxidant. This is what is called a ferryl oxo species. Ferryl oxo species. Okay. So, now what will be the, now this nitrogen will attack here in a 4-exo trig fashion. So, the first ring is formed. Okay. The first ring formation has taken place. S Fe double bond O, that is the +4 oxidation level, and this was CO2H and then 2 methyls. Okay. Now, what will happen? This is a superoxidant. What it does, if it sees a hydrogen, if it sees a hydrogen which it can abstract, it can, if it sees a hydrogen nearby, it will abstract that hydrogen, provided the, after the abstraction of the hydrogen, the resulting species, whether it is a radical or cation or anion, that has to be stabilized. Like if it is suppose allyl, I give you some examples. Suppose if you have allyl and if you generate a ferryl oxo, we can actually generate ferryl oxo species. There are techniques by for generating hydrogen peroxide and iron. Fenton's reagent, very famous. Okay. Then Wooden-Fritz reagent. These are something which makes ferryl oxo species. So, the oxygen will abstract the hydrogen usually in a radical fashion. So, the hydrogen that will be abstracted will produce a radical that is stabilized. So, it will not abstract hydrogen from the homoallylic hydrogen. It will abstract the hydrogen from the allylic carbon, because the allylic radical or the cation, if it is cation suppose, or radical, because there was again this debate whether it goes by a radical or it goes by a cation. But to cut the story short, we are not going into big details, but to cut the story short, that this is the mechanism. The oxygen abstracts the hydrogen and the hydrogen forms a radical here. Okay. So, where should I write? Let me erase some of this, otherwise okay. So, now what is going to happen? Let us see. So, after this, so you have the beta-lactam already formed, you have sulfur, you have iron, and you have this CO2H and a methyl a carbon radical. And remember, this is 1 electron shift. So, iron must be having 1 electron back to it. So, iron is now in +3 oxidation level. However, this iron with 1 electron and the carbon 1 electron, they can actually form a bond also. So, it says that they exist as a resonance hybrid of an organometallic compound, where there is a carbon iron bond. So, there are 2 methyls, and also it is a radical character. So, the intermediate is an organometallic compound, but because iron carbon bonds, at least these bonds are not very stable. So, you can think of that they equilibrate between these two structures. Okay. Now, this radical attacks the sulfur and the iron gets back its electron. So, you have this 5-membered ring formation now, that is the penicillin. So, that gives you penicillin, and you see the iron is back to the ferrous state, because it has got back both the electrons which it started. Okay. 1 electron it got back from this when the oxygen attacked the hydrogen in a radical fashion. So, it is a homolytic cleavage. So, 1 electron remains with iron, and the other electron again comes from the breakage of the iron sulfur bond. Okay. So, this is the mechanism. Now, you can say that how do you know that there is a radical that is formed here? It could be a cation, okay, it could be an anion. Now, the radical was proved by, see, any radical reaction is generally EPR spectroscopy or ESR, electron spin resonance spectroscopy. That usually is the gold standard for making this type of statement, that a reaction going through radicals. Okay. However, if the radicals is very short lived and if it is inside an enzyme system, you may not be able to get any EPR signal, and people tried, they did not get any EPR signal. But there are indirect ways of proving that a mechanism is radical based or cation based or anion based. What was this? That if you have a radical which is alpha to a cyclopropyl ring, then what happens? It immediately breaks down into double bond and then this. Okay. This rate of reaction is so fast that when you generate a radical here in this valine moiety, suppose I have a cyclopropyl here in the amino acid. If it is radical, then I am sure that before this attack can take place, it will open up, because it is so fast. This is called a radical clock, and this is a test whether a reaction is going through radical or not. This is called a cyclopropyl radical clock. Okay. So, this was scientists have made this cyclopropyl, and they have shown, because if there is a cyclopropyl, so the radical will now be formed here and you will get a bigger ring. You will get a 4 membered ring beta lactam fused to a much larger ring, and you will get also this. So, some radical will be here, it will be equilibrating between this radical alpha to the cyclopropyl and also the ring open form. Okay. So, that means you will get a 4 membered ring fused with a 5 membered, but with a cyclopropyl at the arm, or you can get a 4 membered ring fused to 1, 2, 3, 4, 5, 6, 7, 8. So, you will get an 8 membered ring fused to a 4 membered ring. Okay. So, so you see that this is one way of getting new penicillins. I will give some examples, because now we know that it is a radical mechanism. So, what you can do, you take different peptides. First of all, you can take an allyl, because the first thing that will come to your mind, that better have an allyl, and this radical will be very stable in that case. So, if you take an allyl, so what you will get? You will get a penicillin which is having allyl moiety, means this is actually vinyl penicillin. Remember, I told you that penicillin skeleton is very difficult to, the pharmacore is very difficult to change. Okay. So, this is the first example of changing the skeleton of the penicillin with different substituents, because synthetically it is not possible. Even if you make the tripeptide, you cannot form this bond is very difficult to form. Okay. So, no synthetic methodology is available. What is available is that the enzyme is really very generous. It accepts lot of different side chains in the amino acid. See, it started, the natural substrate is valine, but you can take allyl, this is called allyl glycine derived peptide, or you can take even the higher homoallyl also, homoallyl glycine. But now, if you take homoallyl glycine, that means your double bond is here now. So, the radical will be formed here and not there. So, you will not get penicillin, you will get cephalosporin, and in fact, that was what was cephalosporin. Okay. And then you can have different substituents where substituted amino acids were taken in place of valine, and this lab where which was basically, this is all work of Sir Jack Baldwin and your E.P. Abraham. I told you about E.P. Abraham. They combined together and then collaborated, and then finally made several, at least 100 penicillins, new penicillins they have made, and many of these penicillins are very effective against gram negative organisms. Okay. So, that is I think that is all about penicillin. I think this mechanism which I told you, this is not the mechanism, sorry, this mechanism which I told you is what is shown in this slide. Okay. So, later on you can take this, see this slide, and where I explained the oxidation level of the iron in a very critical way, okay, because that is what is the important aspect of this mechanism, that what is the status of this iron. If you study later on bioinorganic chemistry, this will be very important, that how to calculate the oxidation level of metal ions, and one example is this iron oxide. Till today, many papers are being published in very well known journals like JACS or Anglo-Anthropy, and where the chemistry of ferriloxo is has been studied, okay, because this is called a superoxidant. So, you can basically activate CH activation is a very important aspect in today's research, and ferriloxo is one way to do the CH activation. Okay. So, now let us very briefly talk about cephalosporin. Penicillin, cephalosporins come from penicillin, okay. And there was one reaction. See, the problem with cephalosporin initially, although it was discovered in the in the late 50s, but the cephalosporin came into the market much later, because you know the penicillin, the 6 APA, that is 6 amino penicillinic acid, was made available by Beecham, and from that amine you can derivatize and get different penicillins. However, they could not get the corresponding 7 amino cephalosporanic acid. If you do not get the amine, then you cannot make different cephalosporins. So, that was a stumbling block in getting cephalosporin, because if you do it synthetically, then it will be very costly. It will be much more costly if you want to do it synthetically. So, cephalosporin took a long time, and finally, there were systems by which you can get 7 amino penicillinic cephalosporanic acid. Okay. And penicillin, after 2 more steps, goes to the cephalosporin. What are those 2 steps? First, the L alpha amino adipoyl goes to the D isomer. So, there is a isomerase or racemase there, which racemizes that is called penicillin N. Penicillin N is the side chain with D configuration, okay. And then that cyclizes by another enzyme system and gives the cephalosporin, but we are not talking about that. We are just because of the, you can, if you want to know, you can study these slides and then check how the cephalosporins are formed. Okay. I told you about the difficulty in getting the cephalosporins. Just maybe a quick brush up before I go into the another type of antibiotics, where the engineering has been done at the genetic level. This was, this was done at the protein level. You are giving different substrates and then getting different penicillins. Only one reaction for organic chemistry point of view, when it was found that cephalosporins were hard to get, Morin, R.B. Morin, there was one chemist, he was working at the company Eli Lilly. It is a very famous company which makes many of these antibiotics. Okay. So, he discovered one reaction, and that is, from penicillin, if you make a sulfoxide, So, you can always oxidize the sulfur and make the sulfoxide and then heat it by a pericyclic reaction like this. This is a 2,3 sigma tropic shift, and then followed by again sulfur cyclization. So, if you do this pericyclic reaction, what you get is sulfur. You have broken the bond here, no? You have broken the bond, a double bond here, sorry, this is methyl and a methyl here, and then you have CO2H and you have SOH, and then very interesting, CH2, this H leaves, this attacks the sulfur and the OH leaves as water. So, what you get is nothing but the cephalosporin. In fact, this was the method in earlier days when 7-aminocephalosporanic acid was not available. So, they were converting the penicillin via this Morin reaction, which is nothing but a 2,3 sigma tropic shift. And because these are 1 that is the bond which is, so 2 3 and this 2. So, this is a 2,3 sigma tropic shift, 2,3 sigma tropic shift, also called the Morin reaction. So, that is the way you can convert cephalosporin penicillin into cephalosporin. That also gave some clue that how cephalosporin can be obtained in the microorganisms from penicillin. Okay. Because if penicillins can be converted by this way, so people thought that maybe this is the mechanism by which penicillin is converted to cephalosporin by the enzymes. However, that is not true. It was not via the sulfoxide. People made this sulfoxide and shown it to the microorganism, but the microorganism refused to accept this as the substrate to make the cephalosporin. But as I told you, cephalosporin, if you are interested, I have the slide, I have already shown the slide to you. You can go back and then see how cephalosporins are formed. Okay. So, that is for the biosynthesis of penicillin and the engineered different different structural variation of penicillin can be made by changing the substrate. Instead of ACV, you take other tripeptides where valine can be changed. Now, you can say, why do not you change the cysteine? If the cysteine is changed by a serine, then you should get an oxapenicillin, but unfortunately, that is not accepted as a substrate. You need the sulfur. Okay. So, not always everything can be accepted, that is not accepted. So, you have to stick to the serine, cysteine, and then valine here there are lot of perturbations you can do. So, different substituted penicillins can be made and have been made. Okay. Okay, thank you. So, just wait for the next session.